Então, pessoal, vamos iniciar mais um Journal Club do FERT Group. Hoje a gente vai falar de testes não invasivos em genética reprodutiva, sobre a colaboração da Progenesis. A gente tem aqui hoje o Dr. Nabil Arrach. É Arrach que fala? Is that right? Arrach?
Arrach, yes, sim.
Okay. So, would you please share your screen, Dr. Nabil, please?
Yes. Okay. Well, thank you. Thank you so much for the invitation. Today, we are going to be talking about non-invasive methods for reproductive genetics. I'm going to start first by talking about what tests have been developed in the past and look at the history a little bit. And then after that, we can talk about the new generation of tests and where these tests can lead us in the future. So if we are talking about the history of PGT, PGT has gone through a lot of transitions from a very basic test, a FISH test, which was a fluorescence test designed to only show a few markers, few chromosomes, to a full chromosome, a full genome assessment with microarray in the early 2000s. And then recently in 2016, we started to see a more robust test using next generation sequencing. And with next generation sequencing, you have the ability to look at the entire genome now with the new projects of whole genome sequencing projects that Progenesis and other companies have engaged to. So as far as non-invasive, when you look at the worldwide usage of PGT, many countries, either in Europe, here represented in green, or in some Asian countries like India, Japan, etc., the non-invasive test has been used more than the biopsy tests. And that is due to multiple factors. One of them is the legislations in certain European countries. You cannot do a biopsy. It's illegal to do biopsy. And so the laws, the regulations on laws in certain countries, and in other circumstances, so you have the limitation cost that countries are not using biopsy because it's more expensive. And so it's more cost saving to look at non-invasive method that could be images, interpretation of embryo images, or it could be a non-invasive genomic test. So when you look at PGT status today, we know that the implantation rates is still limited. It's in the 70% in the best scenarios, while the pregnancy and pregnancy rate is in the 50 to 60%. And so with that, one would ask the question whether why we are not reaching a higher implantation rate and pregnancy rates, and what that is due to, considering that we already test for chromosome copy. So are there other reasons that will make the embryo unfit for implantation and pregnancy? One would think it could be due to the embryo itself. For example, the metabolic activity of the embryo, or it could be due to factors that are outside of the embryo, which is endometrial and patient-related factors. So today we still have not a fully efficient system for biopsy because we have to incur additional stress and additional manipulation of the embryo to obtain that small size of the biopsy that we can test and evaluate the embryos. It is time consuming because we know that biopsy takes time, it takes training, it's more expensive. Plus we have mosaicism, which is a whole different challenge that the industry is facing. And so when you look at all these considerations, you come to the conclusions that the biopsy of an embryo is not an ideal situation. It does give us some of the answers. It does increase some of those rates, but it's not an ideal situation. So as I mentioned, the pregnancy rate as of today in the best scenario is in the 50 to 60% rate. We were one of the first or among the first to publish on non-invasive genomic assay. In this study, we have compared day 3 and day 5 media and how it correlates with the biopsy of an embryo. And we have seen that day 5 has better correlation with the biopsy than day 3. And later on, many other publications were published showing the correlation between media and embryo or media and biopsy. Many of those studies are very small, and so it's really hard to take the rates that they are publishing as a fact because of the limitation of the size of the study. But if you look at big studies like the one we mentioned here in this slide by Rubio 2020, the euploidy and aneuploidy prediction or correlation was in the 78%. And so if you look at the entire body of literature, really is in the mid-70s. The question is that enough to make the genomic test a clinical test? And the straightforward answer is no. A clinical test has to have at least 95% accuracy. And so some of the limitation we see with genomic assay, non-invasive genomic assay, is the fact that it has high rates of false positive and false negatives that are due to maternal contamination. Plus, the consistency in the media is very imperfect. So that, for example, when you look at the abundance of DNA in non-invasive media is not even, is not consistent across all embryos within the same patient and across all patients and across different laboratories. So because of that lack of consistency, it's hard to make it a clinical test. Where the media is coming from, where the DNA is coming from, well, there are multiple sources where the DNAs can come from in the spent media. That could be from the cumulative cells themselves. It could be as a result of apoptosis or necrosis of certain cells in the embryo. You know, when the embryo is developing, there are, for example, in day 5, you have more than 100 cells, and the turnover of those cells, there are situations where the cells may lyse, and then as part of the normal development of an embryo, and those lyse cells will make some of the DNA available in the media. Some of other sources of DNA can be secretion from the embryo, or it could be polar body, or it could be user contamination, introduced by the user themselves. The size of the DNA present in non-invasive in media of an embryo, if you look at the one that are present in spent media, they are in the 700 base pair range, which is really good size and would be an attractive target to analyze if we had a perfect method. How the spent media is being prepared today in some laboratories, whether for research or clinical, you have to do a heavy denudation of the oocytes. Sometimes it's too aggressive to the oocyte to do a heavy denudation. Then you have to culture till embryos. If you are doing group culture, you have to actually spin those embryos individually and day 4. And then you have to wait for 2 days till day 6 to culture the media. So some of the embryo may be ready at day 5. So keeping them at day 6, it's it's a deviation and maybe not an attractive feature to have when it comes to collecting material for testing. So the biggest amount or best quality of DNA obtained was when you compared the day of media change and collecting and the day of sample collection is between day 4 and day 6. So when you look at day 3 and day 5 or continuous culture media from day 1 to day 5, you don't get that much of a DNA that is quality DNA that you would analyze. And so the best scenario would be from day 4, collection on day 6, or in a freeze-thaw exercise, because when you freeze and thaw, you may have additional factors that lead to some DNA being leaking out from the embryo to the external cellular environments. And so ways to improve the media or to improve the accuracy of genomic tests in spent media could be the introduction of markers to identify maternal contamination, maybe changing the culturing system to a system that is favorable to more DNA. And then you have to do some standardization, obviously, across lab and within the lab and across labs to have the same method of collection everywhere. When you look at the rate of PGT non-invasive for PGT standard PGT biopsy, we're looking at 75 versus 95 correlation. Again, this is based on analyzing a DNA is not based on clinical outcome and accuracy in this context, and it is the ability to call a trisomy or a monosomy in a good quality DNA. So PGTA could be in the 95+% versus 75. We've talked about the consistency. Biopsy just offers slightly more consistency in quality. And then you don't have to deal with material contamination as much as spent media. So for this reason, we have decided to, and other groups, to get involved in other non-invasive testing. In this case, we are talking about metabolomic tests. Metabolites, unlike DNA, are always available in the media. You can see them at any time of embryo growth. They're not dependent on the any other factors other than metabolizing the nutrients. And so for this reason, the bottleneck is not, we don't have a bottleneck as we used to have with genomic DNA. The other thing is when we look at metabolomic assay, we are analyzing small molecules. We are analyzing small peptide, amino acid, some of the, we're looking at metabolism of sugar metabolism, glucose metabolism. We're looking at some cofactors like FAD and AD that are involved in respiratory chain and metabolism, glucose metabolism. And we look at certain molecules, stress molecules. We can also look at hormones. But this assay, as of today, is focused on small molecules. And so understanding the metabolic pathway is so important. So as of today, when you grow the embryo till day 5, we have no idea what metabolic pathway are being involved. We think we know, but really we don't have any measurement of that. So imagine if you're able to map what metabolic pathway or escape pathway the embryo is using, you probably have a better idea whether that embryo is passing through a normal metabolic growth and a normal growth in general, right? So, for example, with what we have built so far was the ability to look at these metabolites in media through mass spec. So the way it works is that we take a sample of the media, we add a substrate to it to allow ionization, and then we use laser with the mass spec. We use laser to hit the sample, and we can decide to sample 100 of location within a single drop. So unlike a PGT biopsy test, you only have one biopsy, one lysis, one analysis. You don't have a chance to do a second trial. But with spent media, you have much more volume to work with, but also you can sample hundreds of data points in the same sample. That gives it a really huge advantage because you're looking at a lot of data from a single specimen. And so once the laser is hits the sample, the ions, because the sample now is ionized, the ions will travel through the sensor. So the time it takes the sample to reach the sensor, we call that time to fly, and that time will determine the mass and the charge of the molecule. And so here in this slide on the corner, you're going to see where you have two colors. One is the media itself, and the other one is the embryo, where the media obtained from the embryo. So in this study, we have analyzed 200 samples, of which 150 were subjects to mass spec. Of those samples, 50% were euploid, the other 50 were aneuploid. We also maintained a similar ratio between male and female. And then once we did mass spec on each sample of them, we normalized that data. And then it was subject to machine learning, where part of the data was used for prediction. So the machine learning algorithms are fed the final results. This is euploid, this is aneuploid, and then thousands of peaks per sample. And the machine learning has to digest all that information and come up with certain patterns that are predictive to euploidy or aneuploidy. And the remaining data set was used for testing. In addition, we have used 50 samples to analyze using high-resolution mass spec to identify the exact molecule present in the media. Because at the end, what you want is certain markers that will be predictive of clinical outcome. And so this is the typical profile. In green, you see the media. In blue, you see the spent media coming from the embryo. And obviously, you have different peaks. And then you have certain peaks that have different heights, different intensity. So if you try to do this by hand, it would be virtually impossible. So that's why you have to use a much more powerful statistical tool and machine learning to be able to dive through that complexity. And this is just an example of how machine learning model would work. And so we have tried many algorithms. We've seen compared the accuracy of prediction. So the best model was giving us about 90% accuracy, meaning that we could predict if an embryo is euploid or aneuploid by 90%. And but this is based on a small size, obviously, and we have to look at what this accuracy would look like in a bigger study. This is a heat map where in x-axis you have the samples, and in y-axis you have the sample ID, all the embryos, and on the x-axis you have the peaks. And you can see that certain peaks are present in all samples in dark brown color, and some peaks are present only in certain samples, but not in all samples. This gives you an idea about the diversity of the patterns when you're comparing multiple samples. When you look at the distance between two samples, and when I say distance, I mean from a machine learning perspective, how different a euploid and an aneuploid are in the eye of the machine learning. What we see is that in purple, you see the abnormal, the aneuploid. In green, you see the euploid, and you see that they are clustered into two different categories, right? And then the mosaics are in red, completely different category. However, you see that some of the purple dots are also classified within the normal. They are abnormal according to genomics, but normal according to metabolomic, and vice versa. Some of the candidates are supposed to be normal according to genomics, but they are clustered within the abnormal according to metabolomic. So what that tells us is that maybe we are now looking at those candidates that we will transfer based on euploidy that may not be the best candidate. They may be metabolically unfit. We don't have enough evidence to say, okay, these embryos are in fact metabolically deficient, but this is an initial observation that we're using to dive into this dilemma so far. Okay, so so far we have achieved 90% accuracy in our prediction. If you're looking at PGT non-invasive versus conventional PGT in green with biopsy, and then the NEMA in red, based on the 90% correlation that we see or prediction value that we see, it looks like the NEMA is going to be better than the non-invasive genomics, but not as good as biopsy. But perhaps if we combine the non-invasive genomic with the non-invasive metabolomic, we may have a superior test. So the next phase that we are enrolling multiple centers now to start the next research study where we are looking at prediction of implantation and pregnancy. So instead of predicting euploidy, we wanted to predict implantation and pregnancy. And we wanted to be able to also analyze those embryos that lead to biochemical, because the biochemical losses are typically euploid embryos that miscarry early stage, and then perhaps not because of their genomic makeup, but because of their metabolomic activity. So the study looks like this. Samples are maybe grown in group, in a group culture or single embryo culture. It really doesn't matter. But on day 4, this embryo will be isolated and cultured individually. And then on day 5 or day 6, depend on the day of the biopsy, we will collect that media and we will run machine learning, we will run mass spec and machine learning on it. And then we're going to look at the data versus the clinical outcome. And then the same way, we're going to split the data into one set where it is used to train the algorithms to predict euploidy, to predict implantation and pregnancy, and the other set of data to do a blind test to see if our algorithms are accurate enough to predict. So there are other forms of non-invasive, not just genomic metabolomic. You can also add images. You can add time-lapse images or static images of embryos, and you can combine that with genomic and metabolomic. There are, in fact, they're synergistic. There's a certain synergy between all this data type. So you can evaluate an embryo from a developmental stage, developmental aspects, or you can also evaluate that embryo from metabolomic and from genomic. And there are a lot of studies that have used time-lapse and static images, and the correlation is in the 70%. So if you combine other contexts like metabolomic or genomic or both, then you're likely to have a better prediction. So the reports that we are working on would look like this, where you can look at the rank of the embryo based on genomic, whether that is PGTA with biopsy or non-invasive PGT, the genomic assessment. Then you have metabolomic assessment, where you look at metabolic activity with AI. And then you can add images, time lapse, any other context that on the developmental aspects. And then you have one ranking option where you're trying to transfer the best embryo possible. So this, ideally, if it works well, it will enhance the implantation and pregnancy rates per transfer. So instead of waiting till the second and third transfer, you may get a positive outcome in your first transfer. And so the metabolic profiling can be used beyond embryo screening. What we are currently working on right now is assessing the metabolic profiling of media, the culture media. And then imagine if you're able to profile your media that you are using in the lab and profile different lots, you should be able to tell if there are significant differences between lots. And with machine learning, you can actually predict what lot would give you best results, because you have all that data. You have fertilization rates all the way to blastulation rates, right? And blast usage rates, right? So you can conceivably, you can determine what media composition and dosage of each molecule will give you the best results. If that is possible, then one would be able to re-engineer the media to give you the best results. So the same way you do AI training to predict the best outcome, you can re-engineer that media composition to get an optimum result. So that is the end of my presentation. I would like to thank my team. We have a great team, both on the science and on the commercial side. On the science side, we have AI scientists, bioinformaticians, and mass spec experts that have led these projects. And without them, we would not be here today. Thank you so much. Thank you.
Thank you very much, Dr. Nabil, for the nice presentation. Very, very nice. I think we have some questions. Sarah is asking.
Maybe Fernanda can help us translate.
Yeah, maybe Fernanda. So, Beatriz is saying, thank you very much, Dr. Nabil. It was an excellent lecture. Indeed. Well, I have some questions. Dr. Nabil, do you have any, Sarah is asking something here, but do you guys have any experience in blastocell biopsy or sort of non-invasive?
Yeah, so blastocell, I think blastocell in the non-invasive context, I think it has shown in some studies that has increased slightly the concordance between media and biopsy. In our experience, it's probably about 5% improvement. So if you are relying on media alone, you're probably going to be in the 75%. But if you add the blastocell liquid or fluid, you may increase that to 80%. That's roughly the improvement that we're looking at.
So you think that in the I'm not talking about the metabolomic, talking about the genomic.
Yeah, I'm talking about genomic too.
Yeah, but thinking about genomics, do you think that in the blastocell you may have some more material there in the culture media, or you think that maybe the same amount of DNA?
You get a little bit more DNA, more quality DNA, maybe. But what the studies I have seen, they have a mix of spent media and blastocell mixed, because when you laser the embryo, it starts leaking out the blastocell. So if you give it a little bit of time for recovery, and then you collect the media, you are collecting a media that is a mix between spent media and blastocell, and that seems to enhance slightly the concordance.
Maybe it could have some problem. I think I don't know. I think it's so difficult because of the volume. What to do with that volume you aspirate, and then where you put it? It must be such a small volume to
Yeah. So what labs have done, they will just laser the embryo, and they're not trying to go inside and pull any media. They're just going to leave the embryo sitting there for a minute, and then the embryo will leak. Eventually, will leak. Also, when you do freeze and thaw, there's a lot of leaking out because of the osmosis. There's a lot of leaking outside the material that also seems to enhance the accuracy.
Okay. So yeah, there are some questions here. Can you read it? In the comments. So Sarah is asking in non-invasive PGT, is centrifugation necessary during the sample preparation? It's more a technical question. She's asking if the sample from the media must be centrifugated.
In the metabolomic?
I think she's asking in the genomics.
In the genomic, there's no centrifugation because the DNA is floating there. There are two ways you can go about it. You can try to purify the DNA through beads purification. So beads meaning they use magnetic beads, and then that would or just regular beads for DNA capture beads, and those beads will absorb the DNA, and then you wash, and then you elute and you recover it. So that could be an enrichment step that is being used to obtain concentrated DNA. And the other one, you can just use it as is. You take the media and then you just use a bigger volume, and then you scale up your reaction. But I have seen both methods. Both seem to work.
Yeah. She was asking here about the PCR. I think that's
Yes.
not the.
Yes, but there is no centrifugation assay because if you like the amount of the DNA is so small that if you try to centrifugate, you're gonna lose it.
So, Dr. Jacob, Marcelo Jacob, Fernanda, would you like to translate? You are mute. Yeah.
Fernanda, you're muted.
You're not. Fernanda, you are mute.
Sorry.
Thank you.
So continuing the last question, she also asked what's the best DNA extraction and amplification for non-invasive PGT?
Well, for now, there is really no extraction per se. I think you just take the media and then you lyse it. You lyse it because some of the DNA are still attached to a big DNA structure. So it lyses, it can help. And then you proceed with amplification. There is no such an extraction. But I mentioned earlier, you could purify the DNA using using a bead strategy, and that has also been used. But there is no an actual extraction of the DNA because of that limited amount present there. So you just proceed with amplification straight from the media.
Awesome. So the next question is from Dr. Marcelo Jacobi. He asking about the cost, the comparison of cost between PGTA and NEMA.
So the NEMA is still not commercial. It's still in research stage. So it's free. It's free for those centers who wanted to participate. But when NEMA becomes commercial, it will be the same as a PGT. I think the saving is going to come from the biopsy. The labs are not going to need to do a biopsy. And then also it's going to increase the accuracy because the media has much more volume to work with than a biopsy, because you can do dozens of tests on the media, while on the biopsy you only have one test. So for that reason, you have a second chance and a third chance to work on it. And so I think the accuracy is going to be slightly better or at least consistency in giving results, conclusive results, it may be slightly higher with the NEMA, and even NEMA combined with genomics would be a good approach to. But as far as pricing, I don't think there's going to be any change in pricing, but right now the test is not commercial yet.
And Dr. Nabil, Dr. Marcelo still asks a second question. He asks if the workflow in the lab need to be modified to avoid DNA contamination, or if this question was already resolved.
Yes, so for NEMA, we don't measure DNA. So this way,
for non-invasive PGT.
For non-invasive PGT, contamination comes mostly from the cumulus cells. So the way to avoid that, you have to have some markers, maternal contamination markers, to be able to distinguish between an embryonic material and a material that is coming from cumulus cells. So with those markers, it could be STR, single tandem repeat markers, or it could be SNP markers. If you see that the spent media is matching 100% the cumulus cells, that means it's a contaminant. You're just looking at the same thing. So that will remove contamination, possibility of contamination.
Okay. So there's another question from Beatriz. She's asking for you to kindly share your insights on the prospects of gamete analysis and non-invasive approach of reproductive genetics. So have you any experience, or have you any thoughts about non-invasive? I think she's talking about the non-invasive assessment of the oocyte, probably, right? Is that right, Beatriz? Do oocyte, né? Probably.
Yes, said yes.
Intelligence for
Oocyte?
Oocyte evaluation, né? If you have any insights.
Seria mais a análise do próprio DNA das células germinativas do que dos embriões, né? E já da análise final, nesse caso. Uma forma de analisar os gametas no lugar dos embriões.
So for AI on oocyte, there have been multiple studies. One of them is assessing the oocyte based on images. You just take images, and then you look at the fertilization rates and blastulation rates, and then you train the algorithm to identify from the images to identify the best oocyte, right? So this is one approach. Another approach is more molecular, where there are studies that have looked at genetic markers in cumulus cells to predict the oocytes. So they will profile cumulus cells with multiple markers using RNA and gene expression and all that, or mutation analysis, and then they will determine the quality of the oocyte based on cumulus cells profile, genetic profile. But there are also studies that have looked at oocytes, and they have looked at the progress of the embryo from fertilization all the way to blastulation, and depend how that oocyte does, they would use that to score the oocyte. They would remove male factor, because male factor can be masking the true data. So they are using just donors, sperm donors, or non-male factor sperm providers. And then they are looking at the developmental aspect of the fertilized eggs and correlating that with images of eggs. So there are multiple studies. I think predicting also the likelihood of an oocyte leading to blast, there are calculations that have been made based on all this AI approach. But at the end, the AI would be used for oocyte assessment and for embryo assessments at the same time, could be used.
Thank you very much, Dr. Nabil.
Thank you. Thank you. I think you are muted. Yes.
So Dr. Nabil and Fernanda, a question for you both. Do you think that it's going to be soon that we may have the NEMA for commercial use?
Yeah, I think NEMA will be available probably in less than 6 months, probably 4 or 5 months. We have gone through the first study. Now the second study, we're going to look at different clinics without changing their culturing system. We don't want to change the culture system. We want to just adjust to the laboratory conditions that IVF has. And I think with that, we're going to have a really good assessment of how the NEMA is doing. If we are able to rank the embryos based on metabolic profiling and other type of information, then I think this could be an added value. Whether the clinic wanted to continue to use biopsy or not, it could add value because you can now rank the embryos. So I think in a few months, we should be able to provide this as a solution.
So once again, thank you very much, Dr. Nabil, for the nice presentation. Thank you, Fernanda. Thank you, Progenesis. And
Thank you, Daniela.
Any other lectures in the future.
Thank you so much. Thank you for your time.
Thank you, guys. Obrigada a todo mundo, pessoal. Valeu. Tchau, gente. Beijo. Obrigada.
Tchau.
Thank you very much, guys.
Bye-bye.
See you.
Bye-bye.